Madhav, can you just say something?
Yeah, can you hear me?
Yeah. Okay. We'll wait for just a couple of minutes and then
Yeah,
we'll start.
sure.
Some people are not able to access the link, so we are just resending the links to them.
Oh, okay. Got
So,
it. Yeah.
सर इसके स्पीकर कहाँ पर है? साइड पे होती क्या?
हाँ ये साइड वाला ये मुझे लगा यहाँ
पर है ना? यहाँ
Bonnie, I'm just going to get a glass of water. I'll be back in a minute.
Yeah, sure. आइडिया बना लिया लेट से सब कुछ खत्म होने के बाद जो है नहीं मैं I think Seema ma'am was the only one who joined. She joined, but other than that, yeah. And now Ritamra is there. स्पीकर भी पर स्पीकर Okay. Other people are just joining, but then I think I'll just introduce Madhav and then let's start. We don't
Sure.
want to waste time.
Right.
So I actually happened to meet Madhav accidentally. I mean, among many people I meet, but that was really accidental meeting, and then we associated since last 3 years now. No, Madhav, it's like almost 3 years since we are working together, and it's always every time it's fascinating to see. I mean, often what happens that with clinicians that it's sometimes hard to interact in terms of biological insight. So they are at a different level, and then you are at a different level. So it's always difficult. But with Madhav, it was so easy. The reason why I think you had that postdoctoral experience, which gave you that language that through which you can interact to the basic scientists a little more perfectly. So Madhav, he's associate professor in Department of Nephrology in School of Medicine at Yale, and he's also director of research in kidney transplantation. He's a steering committee member of Human and Translation Immunology program. So about his career, he did his MBBS from Calicut. So, but don't think like I'm also from Kerala. That is why I know him. We never met in that role. In fact, we never met in Malayali community outside here. I tried to call him many times, but he's always busy. So then he did his PG from PGI. So he's an alumni of this place. He did his PG in 2000. 2006, and then he was chief resident later on. Then he did another residency for State University of New York, upstate, in 2010, and then moved to ICANN School of Medicine for a postdoctoral fellowship in 2013. And he was also a Riccardi Miller Transplant Institute Fellow, again in Mount Sinai, and later on he joined AF. Now he's a consultant there, also a active researcher, maintaining a very strong research program on various aspects of this kidney transplant rejection specifically, and which is the area where we are working together. So he's going to show us some exciting data, and it's all to you, Madhav.
Yes, thank you. Thank you. I hope everybody can hear me. Thank you, Bonnie. Thank you, Dr. Rawat, for this opportunity. I mean, it's always great to have anything to do with PGI. I believe that I learned many important things at PGI. I came to PGI way back as a excited medical student who knew some stuff, but I became a doctor when I left PGI, I think. And for this, I'm forever grateful. I think the best of us stayed back at PGI, all of those who are there at PGI and working hard. I think you are the best. The people who left may not have been the best. So with that said, I'm going to share my screen. Can you guys see this?
Yes, yes.
Okay, good, great. So I'll talk about unraveling our experience, unraveling relevant non-HLA genetic mismatches in kidney transplantation, really a bedside to bench approach. I had included the word variants in there, and we have done work on variants that are not mismatches, but I decided to leave that out because at least we could discuss one thing more comprehensively than get sidetracked by the variants. But we've had success in defining some variants and defining the mechanisms of these variants. So those are my disclosures. So I'll talk very briefly about HLA and non-HLA mismatches, about the scales of genome-wide non-HLA donor-recipient mismatches, and how they associate with kidney allograft outcomes in the current paradigm, and how we've tried to use some approaches to discover those non-HLA mismatches that are of disproportionate significance to kidney transplantation, including this one on chromosome 2, which is the LIMS1 GCC2 locus, and in how we try to unravel mechanisms. How do we study mechanisms once we've identified a candidate? So as many of you know, a problem in kidney transplantation is long-term graft survival. This is a famous paper by Dr. Hariharan, and I think this paper's data, even though it's old, pretty much remains. The rates of acute rejection have gone down. Now we are looking at less than 10% acute rejection in the first year, but the rates of graft survival have not proportionately increased. So there is a discordance. We've been able to address acute rejection, but our problem with long-term graft survival persists. Let me, in PGI style, let's talk about just a scenario of two recipients. This is recipient 1 who got a kidney. She's 55 years old. She has end-stage renal disease from diabetes. Her kidney transplant is in 2020. She gets a deceased donor transplant. The donor is a Caucasian 45-year-old male who died from a cerebrovascular accident. And let's look at another recipient who gets the second kidney from the same donor, who's a slightly older African-American female, and her ESRD was from a different cause. They're both equally mismatched. They have 3 mismatches to this donor. They were induced similarly with thymoglobulin. They were maintained similarly with tacrolimus, MMF, and prednisone. They had their goals attained, so they had, they were treated similarly, is what I'm trying to get at. And they did not have rejection at 1 year. However, when we do a surveillance biopsy, which we do at Yale and many other centers, I'm not sure if they do at PGI, recipient 1 has no rejection. She has very little IFTA. IFTA is fibrosis, interstitial fibrosis tubular atrophy. She's grade 1, and she has a reasonably good creatinine of 1.4, whereas this other person, who theoretically is very much the same, has now developed some amount of tubular injury, tubulitis, and has more IFTA, grade 2 IFTA, and her creatinine is higher. So something has started to pull these two patients who started out very similar apart. And so what is it? So keeping that in the background, let's talk about HLA and non-HLA mismatches. So if people who are dealing with kidney transplantation know that we use HLA A, B, and DR mismatches in order for prioritizing organs, not really for excluding organs, only for prioritizing, giving some score. We cannot exclude organs based on mismatches because we just don't have that many organs, and this is probably the case in India as well. But look at these three donors for this recipient. These three donors have the same level of mismatch. For this recipient, each of these donor is a zero mismatch donor. But the level of matching at the A, B, and DR loci are different. So one is a 3 match, one is a 5 match, one is a 6 match. What I'm trying to point out here is that there is some difference between matching and mismatching. And it's mismatching that we use in order to allocate organs or look at what's happening. And this is our work we published in 2017 in Kidney International. We looked at about 100,000 kidney transplants in the US. We looked at only in deceased donor transplants are mismatching, HLA mismatch, every level. So if you have 0 mismatch, you're 0, 1 mismatch, 2 mismatch at the AB, and the maximum you can get is 6 mismatch. Every level of mismatch and every level of match, do they still contribute in adjusted models to graft survival? And as you can see from this very nice curve, it's a stepwise effect. Every level of matching, every level of mismatching, both of them contribute to graft survival, even when you isolate deceased donor transplants. But if you put both of them in the same model, mismatching and matching into the same model, from these forest plots, you can see that really match, which is on the top, is where there's a significant association. When you adjust for match, mismatching loses all of its association. So the effect on graft survival, the association with graft survival is really improved when you use matching. And so why is that? We asked, theoretically, this should not be so much of a problem. Why is that? And there are several potential explanations for this, and I'm not going to go into each of them, but one of the potential explanations is that when you look at higher degrees of matches, are you really picking up co-inheritance of non-HLA genetic variants? Variants that are, for instance, variants that come with ancestry. If you're Caucasian, certain HLA types are more common. If you're Caucasian to Caucasian transplant, then you're more likely to be, in the higher level of matching. And is that what's driving this difference? So we asked this question at the time. The fact that non-HLA, based proteins, based genes drive some, graft outcomes is explained by very simple mouse experiments. If you take a syngenic mouse, so they're both the same kind of MHC, and you transplant male skin into a female mouse, syngenic, versus a female skin into a female mouse, the male skin into a female mouse rejects. So here the MHC is identical, everything is basically identical. The main difference is that they have some genes that are coming from the Y chromosome. So this is the non-HLA based, genetic, coded proteins. And that is, so that's a very simple example that non-HLA genetics does influence graft rejection. And this is a paper in the human context where Dr. Colvin's group looked at both completely HLA identical groups. So they are patients who are HLA identical. So they cannot have HLA mismatch. They're identical. And he showed that some of them go on and develop antibody-mediated rejection. So they have C4D, they have what looks like antibody-mediated rejection. By definition, such rejection must be driven by non-HLA antibodies, since they cannot develop HLA antibodies in this milieu. Their HLA is identical to the donor. So both of these prove that in animal and mouse context, this is still happening. This problem with non-HLA mismatches is still happening. And just to remind everybody, the MHC region is a very small region. When you stretch out the chromosomes like this, it's a very small region on chromosome 6. And there's a lot of variation that's impacted into other parts of the genome that we are not really considering. And the other part, just for the audience, is that we must remember DNA vocabulary. The majority of variation between two individuals comes because of non-coding sequences. Introns or intergenic regions, because they are actually the most prevalent. Only about 1.5% of the whole genome is protein coding. So any variation is mostly between two individuals is mostly non-coding. And when it's at the coding level, we can call variation synonymous when the variation does not induce an amino acid change, are non-synonymous when the variation induces a protein, an amino acid change. So just keeping that as the background, what about what's happening in recent data when you look at genome-wide mismatch signals exclusively coming from the non-HLA and outcomes in kidney transplantation? And so this is a paper was published in the Lancet by Dr. Oberbauer's group, and they did this very nicely. They took a Caucasian to Caucasian cohort. There are about 450 donor-recipient pairs or so, and they looked at graft survival. They looked at the number of mismatches only in protein coding non-synonymous regions. So the donor had to be introducing, you cannot be sure, but most likely the donor was introducing a peptide difference. There was a new peptide coming from the donor into the recipient. And if you simply arrange the number of mismatches in this manner into quartiles, you see that if you are more mismatched at these protein coding non-synonymous mismatches, you tended to have more graft loss, and this is shown here. And a small proportion of them developed non-HLA antibodies. So they had the full range. They developed an immune response that led to graft loss, and this immune response in some of them was characterized by the development of non-HLA antibodies, which basically says that there was a classical adaptive immune response. The recipient was recognizing the donor peptides as non-self because they were structurally different, and they were mounting a classical adaptive immune response against these polymorphic non-HLA peptides. And that makes a lot of sense. And this simply what we talk about in a classical adaptive immune response. An antigen-presenting cell is going to present a peptide to a recipient T cell, and the recipient T cell, if it has a T cell receptor that's able to recognize it, is going to see this as non-self and get activated if there's enough co-stimulation and other things, and culminating in the presence of non-HLA antibodies. And that makes a lot of sense. But things get interesting when you look at some other papers. This is a paper from Mini Sarwar's group, and they looked at a group of people who had rejection versus a group of people who had no rejection. They looked at the donors and recipients. This is a small data set, only 28 donor-recipient pairs were included in this, but they did see that the number of mismatches at non-HLA variants was higher if associated with rejection, and lower when you didn't have rejection. These were significantly different. But very interestingly, these variants that were contributing to this bad outcome were non-coding variants that had regulatory function, not coding variants in this analysis. And that was very surprising. They also localized many of these genes where the variants were located to endothelial cells, so providing two interesting data points from this paper. We also have to ask ourselves, why do kidney transplants fail? This is a paper from the Mayo Clinic. It was published in 2010. They looked at grafts that failed. They went back into the chart, looked at the last biopsy available for each of those patients, and tried to come up with what was the reason that the graft failed. And you could see that in about 30% of the cases, the only thing they found was fibrosis atrophy, just a kind of scarring. The grafts scarred themselves and they failed. There were other etiologies that were clear, but there were a good proportion where the grafts just failed because of fibrosis. And we also know from Dr. Nankival's paper from Sydney that this kind of fibrosis, when you serially biopsy patients, develops early. So this is the CI plus CT score, and you see by 2 years, there's a peak, and then it's kind of flattens out. So this kind of fibrosis develops very early, and a large number of grafts have at least mild or moderate fibrosis by 2 years. So this kind of fibrosis that the grafts develop, they develop early. And this is our own work. We published this in the Lancet a few years ago, and we showed that if you have this level of fibrosis at the 1 year time point, that those grafts that have fibrosis are more likely to fail than the grafts that don't have fibrosis. So not only is this fibrosis important for graft failure, it develops early, and when it develops early at that time point, it's a surrogate for bad things to happen in the future. And this is a paper, actually, I was the reviewer for this paper, and what they looked at gene expression profiling from biopsies that have fibrosis, biopsies that have rejection, and biopsies that have no rejection, stable. And what they showed is that when you just look at fibrosis without rejection and compare and look at the differentially expressed genes versus rejection versus no rejection and differentially expressed genes, there is a significant overlap. So rejection, which is a pure immunological phenomenon, looks a lot transcriptionally like fibrosis. So fibrosis is, is it rejection without overt inflammation? What we are really looking at is there a component of non-self-injury without overt inflammation? And this is our work, which we published a few years ago, and was highlighted by ISN, where we looked at genome-wide non-HLA donor-recipient differences and how they influence renal allograft survival. And so we took, I was fortunate that when I went to Mount Sinai and I wanted to work in the lab, I was able to work with Barbara Murphy and Dr. He, and spend those postdoctoral years. There were, biopsies were being done. There was a prospective study, and it is, I was at the right place at the right time. And so we looked 385 donor-recipient pairs, and we did genome-wide SNP genotyping, and basically got an idea of these people's genomes. And we categorized non-HLA genetic differences on two, different scales. The first scale we used was ancestry. So, you can see in this plot, which, each of these dots is either a donor or a recipient from these 385. All the Caucasians, Caucasian ancestry, are at this blue pole. All the African ancestry are at this black pole. All the East Asian Han Chinese ancestry are at this orange pole. And people who don't clearly fit into these ancestries are in between, which will include a significant proportion of South Asians. I know because I did my own genotype in there. And you could, for every donor and recipient, you could calculate a Euclidean distance, the distance between the donor and the recipient on this plot. And that distance alone was associated with graft failure, suggesting if you're genetically dissimilar, even adjusted for HLA, on the axis of ancestry, you are more prone to have graft failure. And this variation, as I said, is mostly coming from non-HLA intronic regions. However, as you might be able to conceive, if you are Caucasian and your donor is also Caucasian, the Euclidean distance in that case is very small. You're in this blue pole, but your graft can still fail. So this way of computing donor-recipient differences does not capture the full picture. If you're Caucasian to Caucasian, you could be at the same pole, you could still fail. So we try to employ another metric called the proportion of identical by descent score. And if you were more similar, if you're an identical twin, your score would be 1. If you're an identical twin with the donor, your score would be 1. If you were completely dissimilar, your score would be less than 0.1. If you were a sibling, so you're kind of half matched, you would be about 0.5. So you generated the score across the genome for all of our donor-recipient pairs. And surprisingly, such a dissimilarity score, independent of HLA, was associated with fibrosis in the graft. So you start developing fibrosis when you are more dissimilar, regardless of HLA. HLA mismatch was associated with rejection, but this kind of dissimilarity at non-HLA regions was associated with fibrosis. And that level of fibrosis, as I showed before, was associated with graft loss. So this suggested that this genome-wide non-HLA differences, which are basically driven by non-coding regions, are associated with fibrosis, especially, surprisingly, what we saw was vascular intimal fibrosis. Well, one problem was vascular intimal fibrosis was difficult to separate from interstitial and tubular fibrosis because they all go together. But in the adjusted model, it was really vascular intimal fibrosis that seemed to be driving this outcome. This is very interesting to us, and that's why it got highlighted. So what we try to, nothing is true until you show it in a mouse. So we took I'm joking, but we took a mouse model where, these are special mice which are donors. They are, MHC mismatched. So HLA, like an HLA, MHC mismatched to their recipients, BALB-C recipients. If they are donors, they are MHC matched, but non-MHC mismatched to their BLAC6 recipients. And, we use syngenic controls. So we were really trying to understand if you only isolate non-MHC matches in a system such as a mouse, can you see what really happens to the graft? And, as you can see, these are the syngenic controls. They don't lose weight. We terminate the experiment, but they're fine. But these are our non-MHC mismatched, and they do lose some weight, and they show a smaller decline. But the MHC mismatch, they are more florid. They lose their graphs very quickly by about 2.5 weeks. And this is shown here. And they do have renal failures. What in the end, they are developing renal failure. And when you look at their histology, as the pathologists in the audience know, more blue means more fibrosis. Really, the fibrosis scores like CT, CV, IFTA, which is inflammation plus fibrosis, which is kind of a chronic injury marker, CI plus CT, they're all higher in these non-MHC mismatch mouse transplants. So when you isolate a system where you have only non-MHC mismatch, in that situation without immunosuppression, they develop what looks like chronic damage and fibrosis. So it supported our human data. I mean, this we published only as an abstract because these are very challenging experiments to do. And Vasily, who was our surgeon, who was an eye surgeon from Russia, was not here anymore. So when we look at the axes of donor-recipient mismatch, we have to consider the HLA mismatch. We have to consider mismatch from genetic ancestry, which is mostly intronic. We have to consider ancestry orthogonal non-HLA mismatches, like non-synonymous, where protein coding mismatches, and these are exonic, and other things like relatedness, which are non-coding typically. But really, these are all helpful to tell us, okay, there is a risk. But what can we do about it? What is actionable from this? And really, according to us, the most actionable thing from this is to identify regions or loci where there's disproportionate relevance of mismatches. Because such things could be genotyped ahead of time, we could find a targeted therapy for that, etc. So we have to first isolate which are some areas in the genome which carry disproportionate relevance to graft loss. And this is our work, which was published a couple of years ago, and identifying the LIMS1 GC2 region. We used our study cohorts. We had GOCAR, and then we genotyped this other cohort called CTOT. And we have these many patients. We used one as a discovery cohort and one as validation. The main strength of these cohorts, even though they were a little different in many ways, was that we had at least intermediate term graft survival data. These were followed up, so we could try to map out what happened to graft survival. Rejection is a good outcome if everybody's being biopsied, and we know everybody's being biopsied at the same time, then rejection is a good outcome. But when some people are being biopsied and some people are not being biopsied, rejection becomes a trickier outcome, because just because you are not biopsied doesn't mean you don't have rejection. Whereas graft loss is, I think, a more hard outcome. So we tried to use a cohort where graft loss was available. And at every, we defined a genome-wide mismatch. We defined a gene-level mismatch. We isolated, in our GOCAR discovery cohort, we were able to annotate 20,000 genes across the genome. So for each of those genes, we quantitated a gene-level mismatch. And then those are based on a SNP-level mismatch. And the SNP-level mismatch is when one copy of a new SNP, a new allele, is introduced by the donor into the recipient. That's a single mismatch. When two copies are introduced, that's a double mismatch. So we had this kind of framework of how we're going to define these things. And this is our quality control. I don't want to go too much into this, but we wanted to exclude very rare variants, because that could drive throw us a lot of noise and not give signal. So we made sure that at least 5% of the kidney transplants should have the mismatch. Only then we selected them. And this is to show that we developed a genome-wide mismatch score, interquartile normalized genome-wide mismatch score, which was then proportional to both ancestral mismatch and identity by descent. So this one variable was able to capture variation along both these axes. So we used this variable. And here is to show that genome-wide mismatch in this manner, in this forest plot, was associated with graft loss, adjusted for HLA-A-B-DR mismatch, the discovery cohort on top and the validation cohort at the bottom. In adjusted analysis in the validation cohort, the odds ratio is maintained, but the p-value, because it crosses 1, is not there. When you include even other HLA loci, A, B, C, DP, DQ, DR, still there is a significant association. So at the genome-wide scale, there is something going on. This is not new. Like I said, we've shown this before. Other groups have shown it before. If you isolate the HLA, the non-HLA across the genome, these mismatches are going to be important. This is to show we tried to see if synonymous versus non-synonymous versus intronic. We could bring all those differences. But as you can see from these correlation plots, they're very collinear. It is hard to separate them and bring out independent value when you put multiple things into the same model. So could not do this. We only used the genome-wide mismatch score. And then we focused on how do we find gene-level mismatches, which are the genes where mismatches are important. And what we did is we looked at one gene at a time, computed the mismatch on one gene, adjusted for the mismatches coming from the remaining part of the genome, minus the gene, HLA, and other clinical covariates, and did multivariable models trying to see which gene mismatch, individual gene mismatch, associated with graph loss in a Cox model. And we did this so that we don't pick up noise. We did 4 models, double mismatch, as I showed you before, double mismatch in European ancestry, only European to European ancestry, any mismatch, and any mismatch in European to European ancestry. And we thought to ourselves, if genes came up in all 4 models, then those were probably the most robust genes. And surprisingly, as you can see here, only one gene with a p-value of 0.01 from the 20,000 were associated with graph loss independent of HLA regions and present in all 4 models. And this gene that came up for us was LIMS1. It's located on chromosome 2. Other genes close to LIMS1, GCC2 and GCC2AS, they both came up. They're all in the same region in different models, suggesting that this region is very important as a non-HLA region. And we then looked at the total number of mismatches in the LIMS1 gene. We looked at the upper quantile versus the lower quantile, and if you have more mismatches, you tend to have more graph loss, independent in an independent model. And whether this is any mismatch or double mismatch, this was true. So clearly, this region was important in the discovery cohort. We then validated this in a multivariable model in the validation cohort with a smaller sample size. But again, you could see that LIMS1 mismatch, independent of mismatches across the genome, was still associated with bad outcomes in the graft. If you loosen the threshold and try to capture more genes, so we said p-value of 0.05 into 0.01, then you get 23 genes. And these genes are listed here, so they have different targets that are potentially interesting. 3 of those genes are on chromosome 2 in that region. So 3 out of 23, again, pointing to us that this region is very important. And if you make a multivariable Cox model, plotting the mismatches from the 23 genes versus the rest of the genome minus 23 genes, you can see that the 23 genes carry a disproportionate effect on graph loss, suggesting using these 23 genes may give us some importance for graph survival. Now, we went from genome-wide signals to gene-level signals. So what is the gene that's important here? And then when we saw the LIMS1, we were kind of happy that we found a target, but we were kind of sad that somebody published this already in the New England Journal, though they used a completely different approach, which is reassuring because we used a different approach, they used a different approach, and we arrived on the same gene. This is, because there's a good likelihood in such assays to get noise, and you want to be sure about the truth. So if 2 people arrive at it independently, this is more likely to be true. And in this paper, what they showed is that the locus was this particular locus, 893403, within the LIMS1, which is an intronic locus. And really the bad outcome, in this case it was rejection, they could not find an association with graft survival, happened only when a GG recipient got a kidney, a donor kidney. So at that region, there are 2 possibilities for the SNPs. You can be A or G. If an A donor kidney went into a GG recipient, that's when the bad outcome occurred, not otherwise. So the mismatch had a directional quality to it. There was a direction that was important. And they showed that in these A allele donors basically had higher LIMS1 expression, and GG, because it's intronic, so it doesn't change the structure of the protein, but it can change the level of the protein. And they found that A allele donors had higher LIMS1 expression, and GG recipients had anti-LIMS1 antibodies during the rejection episode. I mean, this data was a little questionable. It doesn't explain what is so special about the GG recipient, because the GG recipient has lower expression of the same gene, but not absent expression. So why would they develop antibodies against this? So there were some parts of this paper that were not clear. So we probed this in greater detail. We confirmed the directional association in our cohort 2. Only when a GG recipient gets an A donor kidney do you get a bad survival, and that is the red line here compared to the blue line. So risk versus no risk. And the other thing about our analysis was we were looking at gene level mismatches, and this was a single SNP from all the SNPs in the gene. So we asked, independent of this single SNP, does the gene, this other SNPs in the gene, bring some relevance to graft survival? And indeed, there were 35 SNPs that were independent of this SNP that brought some, weight to graft survival. And very surprisingly, these 30 of the top 30 of this 35 were a single haplotype. They were completely linked. In any population, they were completely linked. So they traveled as a unit. This traveled as a unit and is a haplotype. And that haplotype mismatch in multivariable models was associated with graft loss, independent of this particular SNP. So we had the SNP within LIMS1, but we also found there are other SNPs in LIMS1 that can contribute to this problem. And this is to show those survival data. 1 copy of the haplotype being introduced was worse than 0 copies. 2 copies of the haplotype being introduced was worse than 0 copies. So there was a dose dependence of mismatch. There was also a directionality of mismatch, very similar to what we had seen in the, especially this was clearly visible in European to European transplants. And what is fascinating, and this is from the 1000 Genome Project, and these are ancestral data from different cohorts that's available for the 1000 Genome Project. And you can see this is the prevalence of the alternative allele and the reference allele in both the haplotype and the LIMS1 SNP in different countries. And you can see India is about halfway. We have, half of our people are one allele and half of our people are the other allele. Whereas if you look at Han Chinese, all of them, nearly all of them, are alternative alleles. So in China, in a pure Asian to Asian transplant, this mismatch is unlikely to be important. Everybody is going to be of the same allele type. But in the US itself, in our transplants, this was about 20%, and I think we're going to see that in most other communities. And we saw that, again, the LIMS1 mismatch was not associated with acute rejection, but was really associated with fibrosis, and these are p-value for fibrosis. So either the number of minor alleles of the haplotype in the donor or mismatches were associated with fibrosis. So here was a single mismatch which was associated not with rejection but with fibrosis, which we had been building towards, which was very satisfying. And then, we went on to, so the SNP is intronic, and this is where the mechanism comes into play. The SNP is intronic. So what we are really saying is an intronic SNP in the donor, when the recipient does not have that intronic SNP, creates a problem that leads to graft loss. So how does it do it? How do intronic SNPs create this problem? And the most, the important way they do it is by regulating gene expression, not by creating different structures of genes. They are the same gene, but they make more or less of the gene because they're regulating gene expression. And you can see that here, if you had 0 copies of the G allele, you had the lowest expression of this gene called GCC2. If you had 2 copies, you had the highest expression of this gene called GCC2 in immune cells. And we think, in the context of rejection, in the context of graft survival, on the recipient side, it's really immune cells that are playing a role. Now, the kidney cells are gone. This is the immune reaction against the donor. So in immune cells, these SNPs regulate a gene called GCC2, which had come up but is not. In fact, they don't regulate LIMS1. These SNPs and the haplotype do not regulate LIMS1. So in the recipient side, we do not think LIMS1 has got anything to do with it, because the SNP does not regulate LIMS1 in the recipient immune cells. And then we went on to say this gene, the GCC2 gene, is expressed in regulatory T cells and other T cells. The SNP has most EQTL activity in regulatory T cells, as you can see. So really we went from a genome-wide inference to a gene-level inference, from a gene-level inference to a SNP-level inference, from a SNP-level inference to a culprit gene. What is the gene that's really driving it? And from the gene to the cell type inference. So really, we were able to connect all this with just in silico and clinical data. And, and then we went on to show the mechanism. I don't want to get people caught up in this, but we think this Golgin, it's been reported in multiple papers that this gene called GCC2 is really regulating TGF-beta SMAD signaling by interfering with a protein called mannose-6-phosphate receptor, GCC2 is a Golgi, and it lives in the Golgi apparatus, and it's important for connecting endosomes to the Golgi. So it's important for recycling proteins. So proteins that come to the endosome, they go either to the lysosome or get recycled to the membrane, then come back again. And this kind of recycling is possible only because of M6PRs. And GCC2 is important for that recycling. And if you have more or less of GCC2, which is what the SNP would do, that would influence this M6PR recycling. And M6PR is important for TGF-beta signaling. So we have to make this kind of connection all in silico, which is kind of nice. And so we went from culprit gene inference to mechanism inference. The other thing we saw, and this is because it's a genetic consortium, is this: if you pay attention to the SNPs that are highlighted in red here, the SNPs that are highlighted in red belong to the same haplotype. This is the haplotype of the New England Journal of Medicine SNP, the one that was reported. And this is the New England Journal SNP itself, RS893403. What you see above are attack-seek peaks. When a SNP has regulatory effect, the DNA in that region is open and is susceptible to transposase, which can be detected by ATAC-seek. And if you have more peaks, that means the SNP is more regulatory. But if you draw a line from this SNP, you see that there are not many peaks associated with this SNP itself. Whether this SNP itself is in the regulatory region is in question. But if you look at some of the other SNPs that are linked to it, they are clear. So there is one SNP that's linked to it that is in the LIMS1 regulatory region, and there's a SNP in the GCC2 regulatory region, which is actually in the transcription start site, as you can see from these peaks. So this SNP was identified by us, by the New England Journal of Medicine, but it is not a culprit SNP. It is only tagging the other SNPs. It's telling you that this is part of a haplotype. So it's a culprit, it's not a culprit SNP, it's a tagging SNP. There are other SNPs that are really doing the job because they're haplotype. And the blue SNPs are the new haplotype we described. So I'm not going to go into the CNV details, it's going to be a little bit off base, so we'll leave that. And then it's fine, you can do all this in silico, you can say, okay, this is what it's supposed to do, this is how it's supposed to work, but nobody's going to believe you. You have to show it mechanism-wise. You have to actually take cells, make more or less of the protein, see what the cells do. And we were able to show here that we overexpressed the protein in tubular cells and in lymphoid cells. You can see the Western blot band here suggesting there's more GCC2 when we overexpress it. In that context, there's more mannose-6-phosphate receptor in the membrane fraction. You can see that here. The darker the band, the more the protein. And you can quantify that here, saying when you have more GCC2, you get more mannose-6-phosphate receptor. As I said, it connects to total TGF-beta signaling. And here you can see that when you have more mannose-6-phosphate receptor, you get more active level of TGF-beta in the supernatant. So these mechanistic experiments in vitro, albeit they're in vitro, are connecting our SNP to GCC2, to M6PR, to TGF-beta signaling. And in lymphocytes, when you overexpress GCC2, you start getting more Helios. For those of you who know, regulatory T cells, which are known to suppress inflammation, in order for T cells to become regulatory, they need to make a couple of transcription factors. One of them is famous, called FOXP3. Another one which determines the stability of regulatory T cells is IKZF2, also called Helios. So when you make more GCC2, these lymphoid cells start making more Helios, turning more into a regulatory phenotype. They also start making FOXP3, although this is not significant, and they start making less interferon gamma. So they are taking on a more regulatory role and a less pro-inflammatory role, which is what we would have hypothesized from our work. And this is the opposite side. We knocked down, so we overexpressed GCC2, we knocked down GCC2 and showed the exact opposite of these findings. Then we can believe that this is true. So a complex story. We identified the region. It is in this LIMS1 GC2 region on chromosome 2. There's a haplotype that we pulled apart. These haplotypes, as well as the SNP in the New England Journal, they regulate in immune cells. They regulate the production of GCC2. You have this haplotype, you have the minor allele, you make more GCC2. You have the major allele, you make less GCC2. So what you're really giving is you're giving a kidney that has high GCC2 levels to a donor to a recipient that has low GCC2 levels. So a donor that has high GCC2 is going to a recipient that has low GCC2. And these affect TGF-beta signaling in opposite ways. More GCC2, more TGF-beta signaling in the donor kidney. More TGF-beta signaling in the donor kidney means more fibrosis. You fibrose quicker. Low GCC2 means impaired Treg function. You cannot regulate your immune responses. That means you have more immune responses against the donor kidney. The two together combine, and you can get this kind of fibrosis and graft loss. So this we reported a couple of years ago, which was very nice, because since we found it and other people found it, it's a true region, and we really have to understand what it's doing. Once we understand what it's doing, we can think about how is this implicated in modern transplantation? What can we do with this information? Can we design a test? Can we design a drug? How can we interfere with this pathway? And so we are, that's what we are doing. We are looking at mechanisms and potential applications right now. We're trying to collaborate with other, organ transplants, cardiac organ transplant, and liver organ transplants, to see if we can pull this out, whether these play a role in other solid organ transplants. The Australian group has seen our paper. They collaborate with us already, and they have applied for a grant, and they're going to test this in real time in a prospective cohort funded by Genome Australia. And then, we are working with Bonnie, and I'm very thankful to Bonnie for helping us do this. We're working with Bonnie to see if we can substitute donor DNA, and I don't want to go into the details, but donor DNA is hard to come by in the US. The donors, for instance, the patients we get, the recipients are with us in Connecticut. We are 3,000, 4,000 miles away from California, but the donor may have come from California. So when we get the recipient, we have the donor kidney, the recipient is transplanted. When we see the recipient in clinic, it's very hard to find the donor's DNA. Donor may have been harvested in a peripheral hospital in the middle of the country. They may not have stored the donor DNA. So the donor DNA not being there is a problem for using this information. And we're trying to see if there's a way to circumvent this problem. And ultimately, we want to really understand mechanisms. So we've made some mouse models which will help us actually recapitulate this. We have a mouse that can be a donor that's going to make more GCC2. We just made this mouse. And we have a mouse that will be a recipient that has less GCC2. So we will try to recapitulate this condition in humans and see what is the mechanism, which cells are being affected, does it cause more rejection, does it cause more fibrosis, can we target it, etc. So this is our ongoing work. So hopefully, without boring you all too much, I've tried to explain to you our work on HLA and non-HLA mismatches, how we can look upon scales of genome-wide non-MHC mismatches, and how in the modern paradigm they are associating with allograft outcomes. They're still important. And our approach, where we discovered the lymph node GCC2 locus, and we have some other candidates, about 23 genes that may be very important, and how, what amount of effort it takes to think through how these are causing, graft survival problems. What is the mechanism? What is the cellular level mechanism of this, and how to unravel mechanisms, our template and our success. So that's all I have, and I want to acknowledge Dr. Murphy, who set me on the track, and Dr. He, who set me on the track of working on these things. These are the folks in my lab. You guys will remember Ashwini, some of you. He was there at PGI. He works in our lab now. We just got a Nature Communications paper with Ashwini as co-first author. So we are very happy that he came and joined us. And Zego and Thomas are the people who've helped us with the genetic work, and these are our collaborators and our funding agencies right now. So that's all I have, and I'm happy to take questions. Thank you again for this opportunity.
Hello, Dr. Madhav. This is Dr. Lekha. My question is, apart from LIMS1 and GCC2, are there other proteins or genes which can influence the non-MHC genes or proteins that you have found that may play a role?
Yeah, so that's great. That's a great question. Of course, and the quick answer is yes. There are other proteins and other genes that influence the allograft outcome. It may be as simple as it depends upon what mismatch you actually carry. So this may need to be individualized. Every person may have a different mismatch and a different, that is the worst case scenario, a different protein may be important in each donor recipient pair. This is one possibility. But there are some common threads. The mica, as you know, is a minor histocompatibility antigen, which people have shown anti-mica antibodies tend to associate with bad outcomes.
And apart from
And
MYC
and then the AT1R, the angiotensin 1 receptor antibodies, people have make angiotensin 1 receptor antibodies, and that has been shown to be associated with rejection. So this is a non-HLA antibody associated with rejection. Unfortunately, for AT1R, it's not so clear that the mismatches are important. The antibody may be important, but whether it's a genetic mismatch that drives it, whether how polymorphic it is, this is not clear. But if you have the AT1R antibodies in the setting of rejection, it is certainly a non-HLA antibody. One of the other problems which I left out here is you have to be very careful when you. There's a paper in the AJT. This at the World Transplant Congress. I don't know if you guys attended this a couple of months ago. A group from Europe, the postdocs in Europe, the group is led from Brendan Keating's lab in NYU. They showed a new region, the completely new region, which was associated with bad outcomes, which is again non-coding, intronic. So it's intergenic actually. It's not even in a gene. It's in between. So many regions like that have come up. But are they all important? Are they all equally important? How do they affect the outcome? These are all still need to be worked. I think when we do this kind of work, we have to keep 3 or 4 things in mind. The ancestry of the donor and the recipient. We have to keep that in mind, because that by itself will lead us to discover some things that are not important. So we should adjust for that. We should fix for that. The HLA mismatches we have to fix, and we have to find the best HLA variable that's suitable. If we simply use 2-digit HLA genotyping, maybe that's not good enough. Maybe we have to use high resolution genotyping. Maybe we have to use eplet level matching, and then see, so account for HLA, and then account for clinical factors. And because transplant rejection, transplant failure is complicated. There are not only these genetic factors, there are many environmental post-management factors that influence the outcome. And then fourth, we have to decide the outcome. The outcome, I think very important to look at graft loss as the outcome. If you look at rejection as the outcome, unless you have a really clean phenotype, you might be led astray. So those are some of our problems in discovering these new loci. But the short answer is yes, there are new loci.
Yes, this is very important, because if we have 2 donors and they are similar on HLA mismatch, then we can look into the ethnicity that may contribute for the better survival. So
Absolutely.
Yes.
Or the limbs one mismatch. If you have 2 donors that are identical in HLA, that would be one place to apply the limbs one mismatch. Yes. And if you have HLA plus limbs one mismatch, that may be the donor you don't want to consider in going forward in the future.
Maybe that will not work for the Asian population. We don't know about the Indian population, whether they have this limb span have any role in the Indian population.
Well, the limbs one, I know we don't have the specific role, but the prevalence of the alleles in the Indian population is somewhat similar to the US. So if it's a problem in the US, in the East Asian, Chinese population, it's definitely doesn't play a role. But I think in the Indian population, it's likely to assume that it plays a role.
Thank you so much.
You're welcome. Thank you. Thanks for those questions.
Good evening, Dr. Madhav. Can I make a comment?
Yes, please.
Nice to listen to your talk. It was really enjoyable. We could, connect so many cases with the way you were telling us. So 2 main scenarios I wanted to ask, or maybe, discuss. One thing is we had, once analyzed our all cases where graft nephrectomy had to be done because of graft intolerance after, they were non-functioning. And at that point of time, the result which I would say was majority of them had a vascular injury, So there was, as you were saying, there was early fibrosis and late fibrosis and some vascular injury. So ultimately, when even intolerance happened, it was, taken away by the vascular injury when even our C4Ds were negative,
I see.
right? So that was something very intriguing, and like I was discussing with our transplant surgeons, so they were. So what to do about it? That was a question.
So let me ask you, Dr. Sija, did they have anti-HLA antibodies? Did they have donor
So
specific?
they all were HLA matched.
They were actually matched. I see. I see.
Yeah,
Oh,
that's
wow.
what.
So those samples are very valuable.
Yeah, I know. Ashwini was very much part of that whole exercise, when we did with one of our PDCC students, Dr. Rashmi.
I see.
So he has all the background information and all
Well,
about
maybe
that.
we can connect and see if there's something we can do with those samples and how what kind of, I don't know if you have sera banked for those patients, if you have DNA banked for those patients.
Serum, I'm not sure of all, but maybe we can, we might be having of maybe 50%. I can try, I'll have to see.
Let me
Right?
connect with you. I think that this is potentially an area where we could do. We're working with somebody at Yale, and we have she has a new assay that tries to look at non-HLA antibodies, and we're planning to run a pilot
Okay,
on a few cases right now.
okay.
Possible that if this is clearly, if this is clearly a non-HLA antibody-mediated phenomenon, this may be a good group to include. Let me connect with you, and we can see if there's
Sure.
something that can come out of this. Okay.
And one more scenario, which is on the extreme other end, that we had a very early graft rejection, almost like on the table. We were asked to do a frozen section to look for if there is any rejection. We did look for it, but there were no thrombi, only neutrophilic margination was there, which can happen, immediately early post-transplant when you take a tissue. So we said, we are not sure of rejection. But, when we got a definitive from the same frozen, what we found was the smooth muscles were totally necrotic at that point of time.
I see.
And again, it was a live matched donor, said donor and recipient. So in that case, we thought maybe there are non-HLA, kind of antibodies which have played role. And to my mind, I thought maybe it is smooth muscle antibody which has, caused the damage.
Did they have
But-
muscle antibody beforehand?
No, that was not reported, obviously, because that was not tested only.
Yeah.
But later, the graft nephrectomy had to be done because graft couldn't have been kept inside. And then when we saw the nephrectomy, there were large areas of infarct. Once again, the renal arteries were bad at different level, intralobar and everything. But we stained with smooth muscle antigen that was not really lost in all the arteries. So segmentally, it was gone.
I see.
So I believe if it were anti-SMA antibodies, that should have affected all components or maybe all levels of arteries, or do you believe there could be differential involvement? Because in biopsy, you would get a smaller artery.
Right. I mean, my first question would be, was it a purely vascular event?
Yes,
like
yes, it was.
No, like a
Yeah.
thrombosis, not immunological, just a thrombosis.
No. It was going beyond the lumen of vessel. The smooth muscle of the artery was gone. Arteriole was gone.
And C4D, was there evidence of complement action?
I think it was negative, Nitya. I think it was negative.
I mean,
Because otherwise we would have, considered that kind of a rejection. But C4D turned out to be negative.
is it worth doing immunofluorescence for immunoglobulins and trying to see if they were there?
That we didn't try, but we thought we'll try putting serum of this patient on a normal, smooth muscle and see whether there were anti-smooth muscle antibodies. But then, after doing SMA on the graft nephrectomy, since bigger arteries were all positive, only segmental loss was there. So I'm not sure whether this kind of exercise is really going to help us or not. But maybe it's still worth trying.
The first thing may be to show if there are antibodies involved there, right? So there's IgG deposition a lot in that area, then we would feel more confident that this is an antibody-mediated phenomenon.
But necrotic tissue,
Yeah, I see.
if I put immunoglobulin, it will stain. So
Yeah. The problem, when we
really pathogenic or it's just binding?
When you try to put antibodies on cells or tissue, you have to be really careful about the background.
Yes, yes.
Get a lot of signal, and you have to have the appropriate controls and really make sure that this is a true signal before interpreting it. This has been our experience, especially in mice, that there's a lot of binding. If you put it on tissues, there's a lot of binding. I don't know. It sounds very interesting and a potential mechanism, that could be uncovered there. Yeah.
So Dr. Lekha's lab, they did try, discovering other antibodies in that particular patient. But in that panel also, I didn't find any anti-smooth muscle antigens,
Oh, so are they using one of the commercially available panels?
I think Lekha can answer that better. Lekha, are you around? Okay.
The commercially available.
So Biman, that panel.
There are two panels available, at least in the US. I don't know where they are. One is coming from Werfen in Germany, and one is coming from One Lambda. Both of them, actually, when we do this pilot, we will have data from that. Again, it's not clear. Nobody has been able to convincingly show that we are detecting meaningful antibodies. They're detecting antibodies, but are they detecting antibodies that are meaningful? It's not clear yet for these panels. Which is why I think we need a different assay. We need a different assay to do that. We can talk more on that.
Okay, yeah. So maybe I'll write to you and send you some pictures so that you get that confidence of what I'm trying to ask you.
Yes.
So that so we can do it that way. Thank you, Dr. Madhav. It was really great interacting with you. Thank you.
Hello, ma'am. Seema here. So this is a case in which, I'm listening to the whole talk. Actually, we did this applied by this Immucor Werfen kit, non-HLA antibody, but 60 antibodies we applied in this case.
Yeah.
And more than 40%, I think 60% of the antibodies, they were flashing. Non-HLA coming out to be positive in this case.
But Dr. Madhav, my concern was those antibodies were like vimentin, nectin, which are very non-specific, In any necrotic tissue, it would come out. So to implicate those in that rejection was my concern. Unless they were smooth muscle specific.
I would, it's true. The Werfen kit may not be, like I said, I don't think anybody's been able to clearly show that, the antibodies that you see on the Werfen kit are truly associated with bad outcomes. There's a lot of noise. You have to look at the baseline sera in each individual. You should look at the sera before they got the transplant and after they got the transplant. So you have
we have done both comparative pre and post in this case.
How does it look in this particular patient? Are there
definitely post transplant the MFIs were much higher when they were done pre-transplant, but they were elevated pre-transplant as well.
So the same candidates were elevated pre-transplant as well.
Yes. 2-3 came up more.
And what about some controls? What about some controls? Do you have some controls where they didn't have this problem and you did the same assay and there's no...
No. We specially put up this for this non-HLA antibody for this case only. Pre and post both together.
That's the kind of problem that we need to...
Yeah, we need to know the background, how it is in general, and then if in this particular case it was out of proportion, then we could have taken it, as being significant. Yeah. Without having done that, I don't think we can really, associate.
Yeah. Okay.
But that essay, Dr. Madhav, that you mentioned, pilot study that you are employed with right now, that will be interesting in such cases.
So we'll see. We're waiting. We ran a pilot on our mice, and it does work. She's reported this in autoimmune diseases. We're trying to see if we can use it. It's an IgG, proteome-wide IgG. So let's see. Let's talk with Dr. Baseja and see where we are.
Okay.
Thank you, Dr. Madhav.
Thank you.
Good evening, Dr. Madhav. I have one question. How the GCC2 overexpression in the donor or in the grafted kidney can affect the properties of the immune cells of the recipient, because the recipient immune cells are going to actually decide whether the graft is going to stay or it is going to be rejected or some kind of inflammation or fibrosis is going to happen. So can you elaborate on that?
Yeah, that's great. Excellent question. That is an excellent question. What we know is that it's only in this combination that badness happens, right? So if a normal GCC2 donor kidney comes into a low GCC2 recipient, you don't get the bad outcome. It only happens when there's more GCC2 in the donor kidney. So something about having more GCC2 is critical for the phenotype. In other words, the immune system, the low GCC2 problem in the recipient is always there, regardless of what kidney he gets. But only when that high GCC2 kidney comes in does he get the problem. So the high GCC2 is definitely causing a problem. To answer, so what problem is it causing? Is it, we think of this as, there are two possibilities, and they may not be mutually exclusive, they may be happening together. One is that having more GCC2 in the kidney could specifically initiate a break in tolerance to GCC2 in a patient who has low GCC2. So, you come with more GCC2, now your T cells who are not familiar with high GCC2 may start seeing this as an antigen and mount an anti-GCC2 focused response. So break in tolerance because the kidney is bringing high GCC2. That's one possibility. The second possibility is that regardless of what the immune cells do, having more GCC2 in the kidney causes the kidney cells to respond to the same amount of injury in a worse fashion. So you have the immune response, a normal tubular cell would proliferate, would try to come back when it gets injured, would try to come back and not fibrose. But what if the GCC2 extra tubular cell, the cell which has more GCC2, cannot come back fully, or fibrosis more, then that's enough for damaging the kidney. So it could either be because GCC2 induces a break in tolerance in the recipient, because there's more GCC2 coming on the or GCC2 in the response to injury of that kidney is aggravated. And it could be both are to figure that out. That's a great question. We're trying to address in our mouse.
So can we imply, by what you're saying, can we imply that donor immune cells, modulation of the donor immune cell pathway, pollinate by the high of GCC2?
Well, donor immune cells. So
that higher GCC2 properties of the
yeah, I think donor immune cells, theoretically possible that donor immune cells, but if you look at recent data, are not long lasting except for macrophages. But if you really look at things like lymphocyte RNA data, there's very few from the donor after about a month. So if they have an effect, it's going to I don't think it's that important. I would be more likely to blame the resident cells like cells or fibroblasts or things.
Will there be some role of TGF beta, because known to suppress? Can there be role because it promotes?
So on the side, this is exactly what we think is happening, right? When you have the low GCC2 expression, you have a low TGF beta state. Less TGF beta, you are more, like you pointed out. So on the recipient side, what happens to the recipient may be more revved up. I didn't show this to you, but when you take a mouse, knockout, no GCC2, revved up immune system. System where GCC2 is made, regardless, no antigen there, just there is immune system. So in the does cause dysregulation, TGF beta. We think it's via TGF beta. That side is beta. On the donor side, at all, having more GCC2, more TGF beta would suppress the donor immune cells, right? Yeah, that role I think is probably trivial. Yeah, it's probably trivial because the donor kidney carries some immune cells, but like I said, by a month or two, donor immune cells are basically gone. There's very few left. We know that now in much greater detail from single-cell RNA data, where they passed apart the donor versus the recipient lymphocytes in kidney tissues, during the acute phase and the chronic phase. And really, after maybe a couple of months, there's very few lymphocytes. So they're not involved. I don't think they can be involved. It's more likely that the extra TGF beta has a problem for kidney endogenous, kidney epithelial cells, like tubular cells and fibroblasts. So I hope you know, maybe that's now clear to you. Yeah.
Thank you so much.
Okay. Great question. Excellent question.
Thank you.
Okay.
Any other questions? Okay, I think.
Okay.
So thank you so
Good.
much, Madhav. So I hope to see you next week.
Yeah, let's see you back, Bonnie. We need to move our work forward.
Thank you so
Okay.
much.
Thank you very much. Thank you very much. Great to
Bye-bye.
talk. Yeah.
We are starting at 6 o'clock in the morning.